Good afternoon, students. Today we are going to study Leishmania. Leishmania belongs to the kingdom Protista. It is a protozoan and blood and tissue species. That is, it belongs to the class Zoomastigophora, and the species are seen as hemoflagellates. In hemoflagellates, we have already learned Trypanosoma brucei and cruzi. Now we are seeing the third one, that is Leishmania. Now, the genus Leishmania is named after Sir William Leishman. He had discovered the Leishmania donovani in the spleen smear of soldiers who died of fever at Dum Dum in India. The disease is also known locally as Dum Dum fever or Kala Azar. Now, Charles Donovan, he also simultaneously started observing the symptoms of Kala Azar in the patients, and he had also published his discovery just few weeks after Leishman. He had been examining the parasite using Leishman stain, and he had observed the mastigote forms and he had named them as Leishman bodies. Now, classification of Leishmania. This parasite belongs to phylum Sarcomastigophora, order Kinetoplastida, family Trypanosomatidae, and genus is Leishmania. The members of genus Leishmania pass their life cycle in two hosts, that is, mammalian vertebrate host and the insect vector female sandfly. In humans and the other vertebrates host, they live and multiply within the macrophages and other phagocytic cells, where they occur exclusively in the mastigote form. The hosts, two hosts are required to complete the life cycle, as I told you. The definite host is man, intermediate host is female sandfly. Reservoir of infection can be dog. In China and Brazil, you will see dog as the reservoir. In India, man is the reservoir. In Africa, rodents. And in Russia, it is jackals. The habitat of this parasite is reticuloendothelial system of vertebrates, that is when the host is man, dog, and hamster. The morphology of Leishmania is that it occurs in two forms or two stages, a mastigote stage and promastigote stage. You can see in the figure, this is promastigote and this is a mastigote phase, where it is oval one and it is a longitudinal. Now, a mastigote stage is a flagellar stage, and this is found in the RA system of the vertebrates, hosts being man, dog, and hamster. The shape is round to oval. The size of the mastigote is 2 to 4 micrometers along the longitudinal axis and has cell membrane nucleus which is less than 1 micrometer in diameter, round or oval, and situated in the middle of the cell or along the side of the cell membrane, and this develops at 37 degrees. See, these are again, you can see this nucleus, Golgi apparatus, mitochondria, kinetoplast, flagellum, which is not protecting but it is inside the cell. Promastigote stage is a flagellar stage. The habitat is the vector. In this Leishmania, it is sandfly, which is pear-shaped, 5 to 10 micrometers in length by 2 to 3 micrometers in width, and the fully developed ones are long, slender, spindle-shaped, 15 to 20 micrometers by 1 to 2 micrometers. So, see, this is what it is. This is a slender form. You can see that in this, the Golgi apparatus are present, nucleus is present, and flagellum, which was inside in the mastigote form, has come out. Kinetoplast is just below the flagellum from where it starts. So, this is how Leishmania donovani is going to be there in two forms. A mastigote form is present in the vertebrates or the definite host, and the insect host is the promastigote form. Leishmania, parasites and diseases. There are different types of Leishmania that are seen, and you will see that they are causing different types of diseases. Leishmania tropica, Leishmania major, Leishmania ethiopica, Leishmania mexicana. They all cause cutaneous leishmaniasis. Leishmania brasiliensis causes mucocutaneous leishmaniasis, and Leishmania donovani, Leishmania infantum, Leishmania chagassi, they cause visceral leishmaniasis. Now this Leishmania donovani, Leishmania infantum, Leishmania chagassi, they are usually found in the Middle East Africa, Indian subcontinent, and Southeast Asia. They are also found in Central and South America. The Tropica major and Ethiopica, they are causing cutaneous leishmaniasis, and they are usually found in the Middle East, Mediterranean coast, Ethiopia, Africa, and India. Then this Brazilian Leishmania brasiliensis, it is usually seen in the South and Central America, Mexico and Western Peru. These are the places which you are going to find this. Now, leishmaniasis are a group of protozoal disease that is caused by parasite genus Leishmania and transmitted to humans by the bite of female phlebotomine sandfly. Leishmaniasis is a vector-borne disease, and leishmaniasis is a zoonotic disease. And this leishmania is responsible to cause various syndromes in humans like visceral leishmaniasis, which is also called as Kala-Azar, cutaneous leishmaniasis, mucocutaneous leishmaniasis, anthroponotic cutaneous leishmaniasis, zoonotic cutaneous leishmaniasis, post-Kala-Azar dermal leishmaniasis. In India, you see both cutaneous and visceral disease occurs, but visceral is far more important leishmaniasis that is found in India. There is an increasing incidence of disease in India, and zoonotic cutaneous leishmaniasis has been discovered in Rajasthan in 1971. The cases of ACL have been reported from Bikaner city. Now, clinically cutaneous leishmaniasis, it has been typed as Leishmania major. It is a zoonotic cutaneous leishmaniasis, wet lesions with severe reactions. Leishmania tropica is an anthropogenic cutaneous leishmaniasis where you will see dry lesions with minimum ulcerations. Oriental sore is a classical cell-limited ulcers that are seen. Now, Kala-Azar, it is also known as leishmaniasis, is a disease caused by protozoan parasite of the genus Leishmania and transmitted by the bite of sandfly. It is known as Kala-Azar or black fever in India, sandfly disease, dum-dum fever, or it is also known as splenomegaly. So these are the synonyms: Kala-Azar, black fever, sandy fly disease, sandfly disease, dum-dum fever, and tropical splenomegaly. Kala-Azar is caused due to blockage and destruction of RE system of mainly bone marrow, spleen, liver, and lymph node. Parasite multiplication causes leukopenia and thrombocytopenia, glum-bearing. Now, leukopenia, that is this destruction of WBCs. Thrombocytopenia, destruction of platelets. Because of it, there is bleeding attributes that are seen. Hemolysis, autoimmune response. These are also seen in Kala-Azar because the antibodies against the red cells, leukocytes, and platelets are produced. The skin affections are dark pigmented. Therefore, the name has been given as Kala Azar. Indian Kala Azar is considered to be non-zoonotic infection with man as the sole reservoir. Human infection is caused by 21 of 30 species that infect mammals. This include Leishmania donovani, complex with 3 species, Leishmania donovani, Leishmania infantum, and Leishmania chagasi. Kala Azar is an endemic disease in 52 districts. Bihar 31, Jharkhand 4, West Bengal 11, and UP 6. This Indian Kala Azar, it is usually affecting the young children's. The infantile type is causes Mediterranean Leishmaniasis, which is endemic among the children below years 5 and especially seen in North Africa, Middle East, and Northeast China, where dogs and wild animals like foxes, jackals, wolves act as the main reservoirs. It is a zoonosis in Sudan and Leishmania donovani infantum infection is a sporadic and vectors are phlebotic, major, longicuspis, perniciosus, etc. Leishmania donovani archibaldi, it is East African type of leishmaniasis. It is a zoonotic disease found in many rural areas and it also has reservoirs like dogs, mongoose, and wild animals. Sandflies of genus Orientalis and Martinez, they are going to act as a vector. The visceral leishmaniasis is caused by Leishmania donovani and infantum was first characterized in India, where it is also known as Kala Azar, meaning black sickness. This I told you already that they infect the young children and has greater tendency to cause lymphadenopathy. It is a intracellular parasite and the macrophages are infected and the activation of macrophages cause inflammation, and eventually the macrophages are destroyed. Thus, the disease, when it progresses, the individual becomes susceptible. Now, post Kala-Azar dermal leishmaniasis. Post Kala-Azar dermal leishmaniasis is a condition when Leishmania donovani invades skin cells, resides and develops there, and manifests as dermal lesions. Some of the Kala-Azar cases, they will manifest as post Kala-Azar dermal leishmaniasis after a few years of treatment. Recently, it is believed that post Kala-Azar dermal leishmaniasis may even appear without passing through the visceral stage. In this PKDL, you will see that hypopigmented patches are seen on the trunk, extremities, and less frequently you will see them on the faces. You will see erythematous patches on the nose, cheeks, chin, and sometimes you will have a butterfly distribution. That is, on both sides of the nose, here it is both sides of the nose, you will see that there is erythematous patches on the cheeks. Yellowish pink nodules are also seen generally on the skin, mostly face, rarely on the mucus of the tongue and eyes. These are granulomatous, soft, and painless. They can be of various sizes, and this granulomatous nodules, there will be no ulceration. Now, this is visceral leishmaniasis. You can see here how it has been. As I told you, on the trunk and skin, you can see all this. Post Kala-Azar dermal leishmaniasis, all these nodules on the face especially. You can see this. Cutaneous leishmaniasis, oriental sore. So this is the oriental sore. So this is cutaneous leishmaniasis. See here on the hand. See here. here. That is, it is concerned only with the skin. Ulceration is there. So here again, all ulcerations. This is a diffuse leishmaniasis. You can see how the face has been there. Whole thing has been mixed up. That's why it is known as diffuse cutaneous leishmaniasis. Mucocutaneous. See all the mucous membranes, how they are involved. And throat, nose, palate. So leishmania are the intracellular parasites. They infect and divide within the macrophages. About 20 different leishmania parasites have been associated with human infection, and this different leishmania, they do not give any immunity for each other. That is, if Donovani is there, Donovani is having its own immunity. Cross immunity is not seen. For no immunity, if your immunity is there for Donovani, it is not going to protect against infantum. So this is how they don't offer cross immunity against one another. The life cycle is completed in two hosts, vertebrate host, it is a masticote form, that is leishman bodies. Insect, it is promasticote form or flagellar form. Reservoir of infection, dogs, jackal, foxes, rodents and other mammals. Indian Kala Azar is considered as non-zoonotic infection with man as the sole reservoir. It is seen in all age group including infants. Peak age in India is 5 to 9 years. Males are affected twice that of the females. The population movement is between endemic to non-endemic areas, results in spread of the infection. And this infection is more common in the poorest of the poor. That means the socioeconomic status is one of the important factor affecting this disease. The people working in the farming, forestry, mining and fishing, they are at a great risk of being bitten by the sandflies, so they are more affected. Argentipus is a proven vector for Kala Azar, and Papatasi and Sargenti, they are more prone, or they are the causative agents of cutaneous leishmaniasis. Sandflies breed in the cracks and crevices in the soil and buildings, tree holes and caves, etc. The hygiene is also important because overcrowding, ill ventilation and organic matter in the environment facilitate the transmission of this infection. The habits of the flies is mostly nocturnal and only females bite, not the males. Now, mode of transmission. In India, Kala Azar is transmitted from person to person by the bite of the female phlebotomine sandfly. Transmission may take place by contamination of the bite wound or by contact when the insect is crushed during the contact act of the feeding. Through blood transfusion and contaminated syringe and needles. Now transmission. Indian Kala Azar has a unique epidemiological feature of being anthropognotic. Human is the only reservoir of the infection. Female sandflies pick up parasites, that is, a mastigote or Leishman bodies, Leishmaniasis bodies, while feeding the infected human host. And the parasite undergoes morphological changes to become flagellates, that is, promastigote or leptomonad forms. Development and multiplication takes place in the gut of the sandflies and moves to the mouth parts. Healthy human hosts get infection when an infective sandfly vector bites them. So coming to the life cycle. The organism is transmitted by the bite of several species of the blood-feeding sandflies, that is, phlebotomous sandflies, which carries promastigote in the anterior gut of the gut and pharynx. When it gains access to the mononuclear phagocytes, where it transforms into the mastigote and divides until the infected cell ruptures. That is, when this infected sandfly, it is going to bite the human being. Through that bite or through that wound, it is going to transmit its promastigote form into the human being. And this promastigote immediately gets converted into a mastigote forms and reaches the mononuclear phagocytes. This mastigote starts dividing and redividing. They usually divide by longitudinal binary fission, and within the cell they multiply, remultiply, and then when the mononuclear cell is going to rupture because it is fully packed with these mastigotes and they are going to break, and this infected cell, when it ruptures, so many mastigotes are released in the blood and they reinfect the other mononuclear cells. So the released organisms infect the other cells. That is, this mastigotes, they will infect the new macrophages. There also the same thing goes on. Within the cell, they sit and they divide and redivide, and after many divisions, the mononuclear cell is completely filled with this mastigote forms. They will rupture, and that's how the cells go on dividing and dividing and infecting the new cells. The sandfly acquires the organism during the month. So now this human being which was infected, in his whole blood, this mastigotes are going to circulate. When they are circulating and any sandfly comes and sits or bites this human being, this mastigote forms is injected into the sandfly in the blood meal. Now, this amastigote form gets converted into flagellate promastigote form and multiply in the gut until the anterior gut and the pharynx is packed. The whole thing goes on multiplying like it has been multiplying in the mononuclear cell. It multiplies in the anterior gut of the sandfly. And when such infected sandfly, it goes and bites the new host, it is going to ingest the promastigote form into the skin of the host. And the new human being is infected. That is how the whole cycle, that is from promastigote to amastigote, amastigote to promastigote, the whole life cycle is completed. Dogs and rodents are the common reservoirs other than the human being. See here. Sandfly takes the blood meal, injects promastigote stage into the skin. From there, the promastigotes are phagocytosed by the macrophages and the promastigote transform into amastigote inside the macrophages. Amastigotes multiply in cells, include the macrophages of various tissues, and then the sandfly takes the blood meal, ingests the macrophages infected with amastigote, and ingestion of these parasitized cells, there are release of the amastigotes. Then this amastigotes transform into promastigote stage in the midgut of the sandfly, and this promastigote divide in the midgut and migrate to the proboscis, and again the cycle continues. Now when the sandfly bites animal and ingests the blood infected with Leishmania, and then it goes and bites the human being, it is going to release the promastigotes. And this promastigotes, when another sandfly bites the human and ingests the blood infected with Leishmania, the cycle continues when the sandfly bites another human or animal reservoir. Either it has to go there to the animal or to the human being. That is how the life cycle is completed. So again here you can see that is Leishmania infantum, how it is causing visceral leishmaniasis. Clinical types of cutaneous leishmaniasis, that is major this is double. Pathogenesis. Infection ranges from asymptomatic to progressive fully developed Kala Azar. Incubation period is around 2 to 4 months. Symptoms begin with low-grade fever and malaise, which is followed by progressive wasting, anemia, and protrusion of the abdomen from enlarged liver and spleen. Now, if the person is not detected and treated, after 2 to 3 years, the disease becomes fatal. In acute cases, you will see that the patient has chills, fevers of 104 degrees degree Fahrenheit. There is vomiting. Sometimes death may occur within 6 to 12 months. Immediate cause of death is usually an invasion of secondary pathogen that the body is unable to combat. Only then the death will be immediate. Otherwise, it is after 2 to 3 years, it can become fatal. Signs and symptoms: recurrent fever, loss of appetite, pallor, and weight loss with progressive emaciation, weakness, skin becomes dry, thin, and scaly, and hair may be lost. Person show grayish discoloration of the skin of the hands, feet, abdomen, and face, which gives the name Kala Azar, meaning black fever. Splenomegaly, hepatomegaly, lymphadenopathy, anemia develops rapidly, anemia with emaciation you can see here. Gross splenomegaly produces a typical appearance of the patient. In cutaneous leishmaniasis, you will see the smear shows LD bodies. See, these are the small LD bodies that are seen. See, here, 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 here. Okay, these are the LD bodies that are seen. The biopsy of this, if you see, you take out the tissue, stain it, culture it in the medium. For premastigote forms, you are going to find them. LD bodies here, you can see demonstrated in the buffy preparations of the blood and bone marrow aspirates. The aspirate taken from large lymph nodes show parasites in 60% of the cases. You can see. These are the parasites. See here. Yes. And they inside also. Okay. Visceral leishmaniasis diagnosis, parasitological diagnosis. Methods you will see: bone marrow aspirate, splenic aspirate, lymph node tissue biopsy. All this I have shown you earlier. Okay. See bone marrow aspiration, and in bone marrow how you can see this. These are the mastigotes that can be seen in the bone marrow. ELISA is an important serodiagnostic tool for leishmaniasis. It is highly sensitive test. Its specificity depends upon the antigen that has been used. Treatment: pentavalent antimony pentostam is given, and amphotericin B is the drug of choice. The treatment of complications should be done with treatment for anemia, treatment for bleeding, treatment for infections.